Well, we've started some fundamental studies looking at the effect of the terminal amino acid groups and their chemistry effects on self-assembly. RADA4, 100% solution at 1% weight per volume in water, showed the characteristic beta sheet formation profile using CD as we see here. We see for a serine-terminated RADA4 sequence a similar, in fact very similar, peak distribution showing beta sheet formation. However, when we tried the same thing with a lysine-terminated RADA4, we saw absolutely no beta sheet formation at 100% solution, so we had to dilute it down to 25% RADA4K5 and 75% RADA4, where we started to see the onset again of this beta sheet peak, which is an indicator of nanofiber formation. So we do see that there is an end group effect that occurs and does regulate how the nanofiber forms. We took these materials and looked at platelet activation using two different markers. You can see here it's a little bit tight, but we did three different donors. You can see that all of these ones basically were the same as the background control. They had little to no increase in platelet activation. But you can see as soon as we added the K5 system, we had, in general, a dose-responsive effect on platelet activation for both markers. Donor number 1 was a little bit higher than expected at lower concentrations, but the other two sort of followed this profile. So it gives us an understanding that there's a peptide-specific dose-responsive event that's activating platelets. Conversely, we've also done C3A activation, and we saw that this was largely activated by the S5 constructs due to the chemistry of the serine with the free hydroxyl group, most likely. However, surprisingly, we didn't see that with the K5, but yet the K5 substrates activated platelets significantly more than the S5 group. So that just gives us an indication that when designing these materials, we have to be careful about the content of the terminal amino acid sequences. So for us, the next steps are to engineer enzymatically cleavable peptide domains to characterize the self-assembly potential of these nanofibers, characterize the enzymatic cleavage as a function of the peptide properties. Right now, we've struck a collaboration with a Korean immunologist to look at the immunogenicity of these engineered constructs. And that's a 3-year project that starts in January. And then we have to maybe examine some more of this platelet activation result that was sort of unexpected. Specifically, the potential of adding platelet activating ligands to the self-assembling domain, which may give us the ability to target diagnostic and therapeutic applications. So on this stream of thought, we could use this same platform to look at specifically activating platelets using these hydrogels, which may augment clot formation via direct stimulation of the platelets at the wound site. We might be able to use this to screen platelet disorders using engineered peptides for activating specific platelet pathways, which might give us an indication of which pathways are working, which ones are not for a specific patient. And then tissue engineering scaffolds, whereby we're incorporating cell-specific ligands so as to encourage cellular growth. And this is a collaboration that's been struck with some people in the medical center at the U of A.